Okay, perfect. Okay, so for number 1, okay, this question concerns a human disease called Angelman syndrome, which displays genomic imprinting. In the case of Angelman syndrome, the imprint silences the paternally inherited alleles. Suppose the recessive is the following cross is done. What are the genotypes and phenotypes of the progeny?
So
okay. I got for the genotypes, I got a 1:1 ratio, which is, the normal, like if you cross 2 heterozygous, you get the 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. But then for the phenotypes, I got like a 1:1 ratio for diseased and non-diseased.
Yeah, that's what I got too.
Okay. Okay, let me see if I have, because I think we need to show.
I can like show my paper how I did it to like the camera. If that's easy.
Oh, do you have it written out on your paper?
Yeah, I just have it like, yeah. Yeah, I can show it to the camera if that's like easier for now.
Sorry, I didn't get that. I think either my internet was cutting out or your internet was cutting out.
I can like show it to the camera, like what I did.
Okay, yeah, that's perfect.
I mean, I just like, it's pretty self-explanatory. I don't know if you can see, but I just like did a little like heterozygous cross, and I highlighted, can you guys hear me?
Yeah.
Okay, so I highlighted the alleles that would be silenced just so I could like see it easier, which is why there's like random orange. But yeah, I just highlighted it just so I could see like what allele would be like turned off or whatever. So yeah.
Is she cutting out to anyone else?
Yeah.
Really?
Because then it just might be my internet. Oh, yeah, okay.
Okay.
Everyone's cutting out a little bit to me.
Okay, do you want to go to like part B?
Yes. Okay, I finally have it open. My gosh, that took forever. Okay, so part B, this question concerns the situation presented in part A. The following cross is done heterozygous male with homozygous recessive female. What are the genotypes and phenotypes of the progeny? So I got 1 heterozygous to 1 homozygous recessive for the genotype.
Yeah, that's what I got.
And then 100% Angelman syndrome for the phenotypes.
Wait, 100% with the disease?
Yes.
Oh, yeah, duh. Wait, what did I do? Okay. Yeah. Okay.
So I can show my paper.
Okay.
Let's see. Okay, can you guys see that?
Yeah.
Okay, so that's what I did for B. I just did that cross, and it is pretty self-explanatory, like you said last time. Okay, now I don't know.
Answers for C and D that you got as well, just so you know.
Oh, okay, wait, let me, I like can't see my screen for some reason. Oh, there we go. Is it back to my face?
Yes.
Yeah. Okay. So for C, suppose a woman has triple X syndrome, 3 X chromosomes, however, I don't know how to say that, X6 have been deleted from 2 of the X chromosomes in each of her cells. How many bar bodies are found in each of this woman's cells? And so,
0.
yeah, I got 0. I can just go ahead and like share share that work the whole time.
If you don't mind.
Yeah,
That works.
okay.
So
Yeah.
how many did you get?
0.
I got 0.
You got 0?
Yeah, because for the X inactivation, there has to be 2 or more X6 present in the cell
Mhm.
to initiate X inactivation.
Perfect. And as we're doing this, are we supposed to be like showing all the parts of how we did it or our thoughts behind it and stuff?
I
No.
think like
Oh.
we just kind of have to explain how we got there for each question.
Okay.
And then, oh, well, like not on your individual one that you turn in. Is that what you're asking?
Well, no, in the video.
Oh yeah, in the video, I think we just like have to explain how we got it, but in the one you turn in, it's just answers, I think.
Yeah.
And then D, suppose you isolate a cell from a female cat with 2 X chromosomes. However, the T6 genes on each X chromosome are non-functional, while the XS genes on each X chromosome are functional. How many bar bodies are expected to be found in this cell?
I got 2.
Yes, I got 2 as well.
Yep, same.
Sounds good.
Okay. And then for 2, it's just fill in the blanks. So I guess we can just go through. For A, I got the SRY
Yeah.
as an example of a holoandric gene.
That's only on the Y chromosome, right?
Yes.
Okay.
Yeah,
Right.
I'm pretty sure. And then B, I got MIC2 or MIC2, however you say it, is an example of a gene found on both the X and Y chromosomes.
Mm-hmm.
Mm-hmm.
Okay, and then C, cross is made between, I'm not even going to try that, male mouse and a female mouse. What are the phenotypes of the offspring? And I got
75% normal.
You got 75%?
Yeah, what did you get?
I got 1 normal to 1 dwarf body. Because both of the parents are heterozygous, but it what's it called?
Silence the mom's allele?
Yeah, it silences the maternal allele. So the phenotypes would just be like a split of the dads. And so half of his are dominant and half of his are recessive.
Okay.
Mm-hmm.
Okay.
That was my understanding of it.
Same here. So 50-50.
Okay. Then D I got sex influenced pattern of inheritance. An allele can be dominant in one sex while the same allele is recessive in the other sex.
Yeah.
Okay. And then E, I got genomic printing. Imprinting is an epigenetic process that occurs during gamete formation, while dosage compensation is an epigenetic process that occurs during embryogenesis.
Okay. Yeah.
Yeah, I got that from the notes.
Mm-hmm.
I think it's like straight from the teacher's notes.
Yeah.
And then F, I got Xist and T6.
Me too.
Okay. And then G, I got autosomes and then chromosomes. Or X chromosomes.
That's what I got too. Okay.
Okay, so are we all on the same page through 2, through all the fill in the blank?
Yep.
Yes, it all sounds good.
Okay, sounds good. Then starting at 3, a woman's father has a deficiency in X-linked recessive disorder. The woman's mother is homozygous and is unaffected. So what is the genotype of the woman's father? What is the genotype of the woman's mother? So knowing that we knew that he was recessive, right? So recessive X and Y, and then she was homozygous and unaffected, so she was homozygous dominant.
Those are all I got what you got.
Okay. And then for B, if the woman, so that's like the child of those 2 that we were just talking about. If the woman has children with a man who does not have OTD, what is the chance that their first child will be a son with OTD? So in the previous question, I found the woman's genotype.
Yeah.
And then I, down here, crossed it with a X-dominant male and got 1/4.
Do you think it would be like a 50% chance for like since there's only like 2 like male offspring, so one of the 2 male offspring would have the disease. So do you think it could be like a 50% chance of that? If that makes sense. I don't know if
I know what you're saying. So I think I would answer 50% if it said like they have a son, what's the chance of their son? But they say, what's the chance that their first child will be a son with OTD?
Okay, yeah, that makes sense. Okay, yeah, I was kind of confused, but then I was like, wait.
No, that makes sense. Yeah, that makes sense.
Okay.
Yeah, I agree.
Okay, so C, identify the genotype of a male with whom the woman could produce a daughter with OTD. So I kind of worked backwards, I guess. Well, I kind of knew from up here at B, I don't know if you can see my cursor, but at B, I kind of knew from this cross that with a dominant male that didn't have OTD, like it didn't make any girls that would have OTD. So I just kind of knew that the male would have to be recessive
Yeah,
in that one. Okay.
Yeah, and wouldn't he have to be recessive anyway? Because for the woman, she would need 2 recessive ones to produce the disease, and the guy can only give 1 X.
Oh yeah, you're right. So like no matter what, if a daughter is going to have OTD, her dad has to have it. You're right.
Yeah, guaranteed.
Okay. So we all got that at C?
Yep,
yeah.
all good.
Okay, and then D, consider the cross described in part C of this question. What are the expected genotypes and phenotypes of the offspring? So I got, just what's written down here. I won't read it all out loud, but you guys can just look at it.
The same.
Okay.
Yeah, 1 to 1.
Okay. Cool. Okay, and then 4, and you said that you wanted to spend a little more time on 4 and 5, right?
Yeah, well, just like it kind of like explain it because I kind of was having issues trying to figure them out, like just like working backwards, I guess, trying to find the parents' phenotypes and genotypes. So
okay, sounds good. We can do that. Okay, so the presence of a beard in some species of goats is determined by an autosomal gene that is dominant in males and recessive in females. So for A, suppose that a cross produces a 3 beard to 1 no beard phenotype ratio among male offspring. What are the genotypes and phenotypes of the parents? Oh yeah, this took like mental power. So I think the way that I did it was I like had to look at male and female at different times. Like I just couldn't do them both at the same time. Cause like for female, the lowercase b is dominant.
Mm-hmm.
And then for male, the uppercase b is dominant. So I just kind of looked at it like that. And in this first cross, I knew that in a mono, whatever it's called, like in a normal Punnett square, to get a 3-to-1 ratio, you have to have
Heterozygous.
yeah, you have to have heterozygous.
Yeah.
Because you want 1/4 to be recessive.
And so I don't know if this helps, but
Oh, wait.
kind of going along the same lines that you said, when they ask you in the question, so it just says the presence of beard is dominant autosomal gene in males and recessive in females. So I normally just pick like one of the genders, and just like she was saying, solve the whole thing and then go back through the females and then just know that it's flipped and then just go through the other side. And so just
Yeah,
separate.
that's the easiest way, I think.
Yeah, two different problems. Agreed.
Okay, so did that make sense?
Yeah.
So it's like kind of the ratio thing. Like if
Mhm.
you, I guess, just like the more you do the Punnett squares, you just kind of like recognize, okay, they want this ratio, and then you just say, okay, is it in favor of male or female to decide if it's going to be a lowercase b or an uppercase b.
So like two heterozygotes will like basically produce like a 3-to-1 or like a 1 to 2-to-1 ratio, right?
Yeah, if it's a mono hybrid cross, meaning
Yeah.
it's just like one trait to one trait like this is,
Yeah.
and it'll be a 3-to-1 ratio. If it's two traits against each other, then it'll be that. Oh, wait. Oh, no, that's the genotype and phenotype. Yeah, so the genotype ratio will be 1 to 2-to-1, and the phenotype ratio will be
3-to-1.
3-to-1. Okay, I was like,
Yep. There you go.
I remember that.
Okay, and then so for the phenotype, once I just figured out that they were both heterozygous, and then I just figured out the phenotype for male heterozygous and for female heterozygous, and they're different.
I think I did that is what I did. I just like wasn't sure. I'm pretty sure.
Yeah, these ones were tricky.
I just like second guessed it. That is
Yeah.
what I did. All right.
Okay, and then for B, suppose that a cross produces a 1 beard to 3 no beard phenotype ratio among female offspring. What are the genotypes and phenotypes of the parents? And I got the exact same thing.
Okay, that's what I did that too, and I was like, wait, I kind of was like sketchy that it was like the exact same thing, so I wasn't sure. I was like, eh.
Yeah, I second guessed that one too. I was like, really? They want me to write down the exact same thing?
Yeah.
I know, right? I think they're just trying to drill in the fact that males and females are reversed.
Yeah.
Yeah, I think so too.
Okay.
Okay, and then for C, suppose a cross between a male with a beard and a female without a beard produces a 1 beard to 1 no beard phenotype ratio among the female offspring. One of the genotypes of the parents. So for this one, I just realized that like only 2 of them could have lowercase b's. So that means only one of these, like only one of the parents' alleles could have a lowercase b because each one of them, anyways, yeah, each one of the parent alleles has 2 alleles in the Punnett square. So that's kind of how I ruled it out.
So another way to think about this one is you're like, okay, so it says suppose cross between a male with a beard. You're like, okay, what is a male with a beard? He has to have a dominant gene, and so you know it either has to be, I don't know, let's say B is for beard. So like a capital B or a heterozygous one, you need a homozygous dominant one, and then Those are your only two options for the guy. Then you go to the girl, and it says that she doesn't have one. So it's the exact same thing. It has to at least have one B that's dominant or two. And so it's just the two options. And from there, you just narrow down your choices to it either have to be homozygous dominant or heterozygous. And then it's easier to work on it from there. So I don't know if that made sense, but.
Yep. Yeah, that made sense.
Yeah, unfortunately, there's only a few options. So even if you tried out all the options, like,
Yeah.
it's not blowing up your paper. So it's you can
Yeah,
try it all
exactly.
out.
That's so true. Okay. And then D, suppose a male and female both which lack beards produce a 1 beard to 1 no beard ratio among the male offspring. What are the genotypes of the parents? And I guess here's D. That was kind of the same thing for me. I was like, okay, I know that I'm looking at male offspring and that the male parent, he didn't have a beard, so he had to be homozygous recessive.
Mm-hmm. Yeah.
So you can just put a little b in every single box, and then you kind of just figure out what you need from there. So we know that we needed a female with no beard, but we know we needed offspring that had beards. So there had to be a dominant allele in there. That one, I think.
Yeah. And then you just figure out if it's 1 dominant or 2 dominant just by figuring it out. Right?
Yep.
Okay.
And then did everyone get that? As their answer, what was it? We were looking at genotypes. So homozygous recessive for the male and heterozygous for the female.
Yeah.
Yeah. Mm-hmm.
Okay. Okay. And then for 5, this one is, oh yeah. So you could write this, oh no, I think you have to write it this way. Oh, I was thinking about last week's. In cats, orange coat color is determined by the B allele and black, or the lowercase b allele, and black coat color is determined by the uppercase b allele. The heterozygous condition produces a phenotype known as tortoise shell. The B and B alleles are X-linked. Suppose you cross an orange male cat to a tortoise shell female cat. What are the genotypes and phenotypes of the progeny? So can you guys see what I have written out here for 5A?
Yeah. Yeah, we can.
Mm-hmm. Yeah.
Okay, that's how I set up the cross.
Oh,
Yeah, I
so
also got that. So.
Okay, and it was 1 to 1 to 1 to 1, right? Yeah.
Mm-hmm. Yeah.
Okay, and then B, this question concerns the trait described in part A. Male tortoiseshell cats are very rare. What is the genotype of a male tortoiseshell cat? And I explained it better on my paper, or like in the Word document, but basically the male has an extra X chromosome.
Mm-hmm.
And one would be dominant and one would be recessive. And then they'd have a Y chromosome as well.
Okay.
Does that make sense?
So okay, well, yeah. So if he's tortoise shell, because it's like the heterozygous genotype, he has to have an extra X chromosome. Basically, there's no other way, right?
Yeah, because you have to be heterozygous. But one of his X chromosomes would be like undergo X inactivation, because just like a female cells do.
Okay.
Okay. So could I try to like sum it up and see if I understand right?
Yeah.
So essentially, this is an X-linked recessive mutation, so it's only traveling with the X chromosome. And so a male genotype is normally XY. And so to have the different shading in the cat, it would have to have two different types of X genes in there. And so when it's growing initially, some of one color is turned off and some of another color is turned off. So since it has two X's, it can be calico or whatever, and then it still has the Y male.
Yeah.
Okay.
Yep, that sounds great. Okay, so does everyone understand that question?
Yeah.
Okay, sounds good. Okay, in the fruit fly, something, don't know how to say that. A mutation produces the scalloped phenotype, irregular margins on the wings. Assume that the scalloped phenotype is the result of an X-linked recessive mutation. Indicate the genotypes and phenotypes of the progeny produced when reciprocal crosses are done. So reciprocal crosses are the ones where you flip the gender and the genotype, right?
Those
the
are kind of confusing me too.
I don't remember. I think you definitely flip the, what was it, male and female?
Yeah, and I think
Yeah.
anything that was dominant is now recessive. So because you're checking it on the X chromosome, you're checking to see if it affects the phenotypes.
Oh, like the it's like if it affects the autosomes or like the sex or like the X chromosome or something, right?
Yeah, so if you guys can see my screen, here for C, I have it like written out. So you can see that the phenotypes changed between these tests. And so I believe, yeah, that means that they are X-linked if the phenotypes change when you do the test cross.
In the first cross, did you do homozygous dominant for the mom, or is it heterozygous? It's heterozygous, right?
It looks heterozygous.
Okay.
Wait,
And then the second cross, it's homozygous recessive, right, for the female?
yeah, the second cross is homozygous recessive. The first cross is, the results are heterozygous for the female, but the cross that I did was dominant, homozygous dominant, because when you do reciprocal crosses, you do homozygous dominant by the recessive male and then homozygous recessive by the dominant male.
Okay.
Does that make sense?
Mm-hmm.
Yeah.
And you just see if it changes. And if it doesn't change the phenotypes, then that means that it was actually an autosomal mutation. It's like on an autosomal chromosome. But because it changed the phenotypes from the first cross to the second cross, that verifies that it's an X-linked recessive mutation.
Yeah. Okay.
Okay, and then for D, this question concerns the scalloped phenotype described in part C. Assume that the scalloped phenotype is the result of an autosomal recessive mutation. Indicate the genotypes and phenotypes of the progeny produced when reciprocal crosses are done. Wait.
So, okay, maybe I'm still confused on reciprocal crosses. So when you do a reciprocal cross, you switch the dominants to recessives and the recessives to dominants?
Yes, and if gender is involved, you switch the gender.
And the point is to see if it's if it affects, or if it's on a sex chromosome or like a regular chromosome, right?
Yes.
Okay.
Here, let me pull up the teacher's notes.
Yeah.
Just like a side note, should we like plan to do this like every Friday at like 6:30?
Yeah, that's fine with me.
Does that work with
Yeah,
everyone?
that's fine. Yeah, it's a good time.
Yeah, sure.
Okay, cool. Just, yeah, me too. Works.
Okay, so where is, oh, it's going to be way down here. Okay, reciprocal crosses are useful genetic tools that can determine if the alleles that control a trait are found on one of the autosomes or on the X chromosome. Says, yeah, if the phenotypes, one way to determine that a gene is X-linked is to do reciprocal crosses. If the phenotypes of the offspring are different in reciprocal crosses, then the gene is X-linked. With an autosomal gene, reciprocal crosses give the same phenotypes among the offspring.
Okay. So a difference means it's X-linked when we're talking about the phenotypes. If they're the same, then it's autosomal.
Yeah.
Yeah, it makes a lot more sense now.
What?
No, I just said it makes a lot more sense now.
Oh, yeah.
Okay, and then I think, was that the last question?
Yeah, that was.
So did you guys get that they were all the same for D? All the progeny were the same?
Yeah.
Yeah.
Okay. Okay, well, I think that's it then.
Wait, hold on. The recent one we did was still C or D?
That was D.
D? How come? I don't know. At the end of question C, it says indicate the genotypes and phenotypes of the progeny produced when reciprocal crosses are done.
Mm-hmm. Here, I'll pull it back up again. Wait. So, for C right here, can you see it?
Yeah, I did that. So we were talking about D recently, right? Or C?
Yeah, we talked about both of them.
Oh, okay.
Yeah, so D was the one that has all heterozygous progeny.
Okay, yeah, that makes sense now.
And then the in C, they were different. I had one female with a normal wing to one male with a normal wing, and then one female with a normal wing to one male with a scalloped wing.
Mm-hmm. Okay.
Okay.
All right, sounds good.
Okay.
Okay, then I'll go ahead and submit this link on the discussion board, and I'll put your guys' names in it.
Okay, thanks.
Okay, cool. Thank you.
Okay. Yeah.
Thank
See you
you.
guys next week.
Yeah,
All
have a
right.
good night. Have a good
See
weekend.
ya.
Yeah.
Bye.